Our last speaker is ChangHwan Lee, and he's going to tell us about non-random Cyclin transcript localization.
It's all right. I know everybody's pretty exhausted and from the long travel and want to throw yourself on the bed and just want to rest, but just bear with me for another 15 minutes, so it's going to be fun. So today, my name is ChangHwan. I meant to delete this, but I'm graduate student from Gladfelter lab at Dartmouth. And today I'll talk about the Cyclin transcript localization and the cell cycle timing variability and how they are linked through an RNA-binding protein and its polyglutamine expansion. So for studying the cell cycle timing variability, we are using model organism called Ashbya, and you can also see that on my t-shirt. This is a multinuclear filamentous fungus and also close relative of a budding yeast. And what makes Ashbya really cool for this research is that you can observe a multiple cell cycle progression and nuclear division within a single cell. So let's look at the movie. You can see that blue light flashes when mitosis occurs. And you can see that mitosis occurs here and there pretty asynchronously, even though they share the common cytoplasm. And the distance between two nuclei is only 3 to 4 microns. So there's got to be some mechanism to control this timing variability. And our research question is that what's the basis of this timing variability in cell cycle? So after a long try, we finally observed the Asynchrony in V5 deletion strain. And this V5 is an analog of a retinoblastoma protein in mammalian cell, and both these are involved in G1S transcription program or pathway. So we are interested in transcription, and especially in G1S stage. From that, we set up a hypothesis that Cyclin and possibly the G1S Cyclin transcripts are heterogeneously localized. So there are different amount of transcripts around each nucleus, and affecting the cell cycle differently, resulting in this kind of cell cycle timing variability. So to test that, we're using single molecule FISH. And what it does is that multiple oligonucleotides taken with the fluorophores hybridize this target mRNA to visualize individual transcripts. So using this on the Ashbya, we've got this beautiful image. So I was so excited, so named it as cactus. So we started with the clean 3 mRNA, and you can sort of see the overall distribution of the transcripts here. And also, when you look at it more closely, then you're going to see that there are different number of transcripts around each nucleus. And we've done this in this single molecule FISH on the other Cyclin transcripts like a clean 1,2, CLIP5,6, and CLIP1,2, and also GPM2,3, which is involved in glycolysis, and this was used as a non-cell cycle related control. So by looking at all these nice images, a lot of questions occur to us, and hopefully to you too, such as like our number of transcripts are different around each nucleus between nuclei, so there's a heterogeneity there, or is there a spatial pattern of localization of the transcripts? And if it is, then the spatial pattern really important for the cell cycle timing variability. To answer these questions, I've written a MATLAB code to automatically detect these transcripts and the nuclei and reconstruct them in 3D in MATLAB. And first, I just put the spheres on each nucleus and count the number of transcripts within those regions of interest. And so that this high degree of variability of number of transcripts through the Cyclin transcripts. And we're wondering that if this variability is just a total random or some kind of systematic variability. So for testing that, again, simulated the random mRNA using MATLAB for 100 times using Poisson distribution, meaning that using the same number of transcripts, like the same mRNA density, and also the same cytoplasmic area in 3D. And like a previous, put the spheres here. And in this time, just see that how many transcripts are different on neighboring nuclei. And also did that on the random mRNA too, and compare these two. And interestingly, we found significant differences between these only in clean 3 mRNAs. So clean 3 mRNA is non-randomly and heterogeneously distributed around each nucleus. And that might be connected with the cell cycle timing variability. But we wanted to see that this is happening throughout the cell with a larger scale, and wanted to use all the hundreds, thousands of images. So for that, we're using the spatial statistics called Ripley's K function. And what it does is that there are two different cases. So in one case, mRNA are clustered, and the other is that it's just a complete spatial random. And you plot the cumulative number of mRNAs against the distance from the center of the nucleus. And in the random, you're going to see this nicely gradual line. But if mRNAs are clustered, then you're going to see a bump here of the random region. And this is indicative of clustering. And we've done the 100 random simulations and actually get the random population. And these dashed lines are 95 confidence interval of this random population, meaning that if any kind of spatial pattern above this random region, that means this clustering is statistically significant. And this analysis is density independent because we use the same number of transcripts and the same space for getting the spatial random from actual mRNA and the random mRNA. And this was from one image, and we have thousands of images. So for easier comparison, we normalize this, just basically flatten this plot out, and you can still see the random region here, and above the random of the spatial pattern means clustering, and below will be dispersion. So with this technique, let's look at the actual data. And surprisingly, we've seen significant amount of clustering in clean-3 mRNA above this random region. And you can compare this spatial pattern with other cyclin transcripts here. And they are nicely fall into this random region and shows some of the dispersion also. And we get this spatial pattern values about the random region and compare these throughout the different cyclin transcripts, and so that only clean-3 is significantly more clustered. So we are, now we know it's true that the clean-3 mRNA are clustered and also heterogeneously localized around each nucleus. Then two kind of questions came up to me. First one is what's the mechanism of this clustering? And the other is that is this clustering important for cell cycle timing variability? So for attacking the first question, we looked at the other players in G1S transition and found this protein called V3. And a little special thing about this V3 is that it contains RRM domain, which is RNA recognition motif. So this RRM can interact and tethers the transcripts like a clean-3 mRNA. So we deleted the whole ORF of this V3 and also just deleted the RRM to disrupt the interaction with the transcript and see how the localization of the transcript changes. So we did a single molecular fish here, and surprisingly, the clean-3 mRNA were no longer clustered like a wild type in V3 deletion and delta RRM2. So this means that V3, and especially its RNA binding function, is essential for clustering the clean-3 mRNA. So going to the next question, does that matter really for the cell cycle timing variability? So we took one of the strain V3 here, and then observe the cell cycle progression here. So let's look at that. These three are dividing at the same time, and some of these two are dividing at the same time. And you're going to see that all of these nuclei are dividing at the same time in one hypha. So we can actually observe the high degree of asynchrony here. And for quantifying this, we developed a statistical tool called the Synchrony index. And what it does is that just compare using the pairwise comparison of the cell cycle stages on neighboring nuclei and tell that how much they are synchronous. So in wild type, it's around 1, and some number above 1 means that they show more synchronous nuclear division, and that means small timing variability in cell cycle. And you can clearly see that V3 deletion and both delta RRM shows a high degree of synchronous nuclear division. So that means that suggests that the clean-3 mRNA clustering is important for cell cycle timing variability. Then how does this V3 functioning on that? So we looked at the localization here and found that the V3 is heterogeneously localized within the cell. So you can see that this kind of nice tubular-like structure or rod-like shape, and you can compare these patterns with the cytoplasmic GFP. And we quantified this So these red lines are the mean values and the blue lines are standard deviation, and you can clearly see the differences between these two. So we are thinking that V3 protein is heterogeneously localized around each nucleus, and since they're tethering the Cl3 mRNA will be heterogeneously localized too, and will affect the cell cycle differently. But in V3 deletion, now you don't have V3, so the transcripts will be just randomly distributed. So will affect the cell cycle pretty similarly, and you will see the really small timing variability in cell cycle. So then what's the basis of this V3 heterogeneity? So since it makes the tubular-like a structure, we thought that it might be on the ER. But on the overlay, as you can see, V3 was not co-localized with ER. So there's got to be some other kind of mechanism to make this clustered. So going back to the protein structure, and we've seen this polyQ region, and you can see a lot of Qs here. And this polyQ is a good indication of the prion-like or amyloid-like a protein. So we deleted this polyQ. I actually substituted this polyQ with a 6HA. So removing this and so that how V3 localization changes. And surprisingly, compared to the wild type, delta polyQ shows this greatly diminished amount of heterogeneity in the V3 localization. And we also quantified this, and you can see the clear differences, and that was significantly different. Then, is this localization is connected and will affect the localization of Cl3 mRNAs too? The answer is yes. So we've done the single molecular fissionness, and you can still see that the, this Cl3 mRNA localization is greatly randomly distributed and not clustered like a wild type, this. And does that affect the cell cycle again too? And there's a connection there. Yeah, you can see that just like a V3 delta polyQ also shows a high degree of asynchronous nuclear division. And that means that the cell cycle timing variability is greatly reduced in delta polyQ too. So that leads us to that it's possible that heterogeneous localization of a V3 generates the clustering of a Cl3 mRNA. And the combination of these will affect the cell cycle timing variability. In other words, so there are going to be V3 proteins again, and there's a polyQ domains here. And due to the polyQ, you're going to see a clustering of V3, different amount around each nucleus. And this will tether the different amount of Cl3 mRNAs around each nucleus too, and will lead the cell cycle progression differently. So there's going to be a variation. And not only in the Ashbya, we think that any kind of unicellular organisms like a budding yeast or the mammalian cell, it's possible that at the division, there are going to be different amount of some regulatory proteins, especially binding some kind of really important and regulatory transcripts here differently located on the two sisters and will affect them. So two sisters will have a different cell fate or cell cycle progression rate even after the division. So with that, I'd like to thank my PI, Amy Gladfelter. She's really one of the greatest mentor and PI I've ever met, and our lab members, and our collaborator, Dr. Mark Borsa. And I'm happy to take any questions.
So what does Cl3 protein do? Presumably it's non-randomly distributed, and that correlates with the cell cycle time.
Yeah,
Have you looked at Cl3?
So, yeah. So G1, we observed that the G1 is highly variable in the cell cycle, but other cell cycle stages like G2 or N phase are pretty small and shows a little differences between them. So I hope I answered your question correctly. But so
did you look at Cl3 protein? That's all I'm asking.
I'm sorry.
Yeah,
so
sorry.
yeah, we've seen the Cl3 localization too, and they show the heterogeneous localization too. So, but I didn't bring up here. So.
What happens to synchrony in cells that overexpress CLIN3 or cells that lack
Oh,
CLIN3?
yeah, that's a good question. So we were wondering about that too. So we overexpressed the V3 too, and also saw that the
Overexpression of CLIN3 or?
oh yeah, both clean.
Okay.
I don't believe we've done the CLIN3 overexpression actually, but we've done the overexpression of V3, and so that also really reduced the amount of timing variability in cell cycle. And we think that that could be possible because also it loses the heterogeneity of V3 because V3 is now everywhere. So you can't really keep the heterogeneity anymore. But we haven't done any overexpression of CLIN3, but we deleted the CLIN3 and also observed the synchronous nuclear division or the less timing variability too. So it's another supporting factor that for this hypothesis.
Hi, is the CLIN3 messenger localization depends on the microtubules, or do you see any co-localization?
That is a good question too, and we don't know if V3 is on the microtubule or some kind of interaction with that. But when we deleted the polyQ, it seems like now the CLIN3 is pretty dispersed. So it is possible that the polyQ is a domain that can interact with some kind of organelle within a cell. But since it's a really strong indicator of the amylio like a protein, we think that just V3 is somehow just clustering by itself, not using any kind of scaffolds. But we don't know.
What is the benefit of the asynchrony to the organism?
So nobody knows, but we believe that, so there are some actually interesting data here too, but we think that if all of them are synchronous and in the same cell cycle stages, if some kind of drug was treated or some kind of really severe environment that inhibits some specific cell cycle stages, then they just have to die, right? But if you have really asynchronous things, then it's possible, it's a chance to just wait a little bit to meet a better kind of environment, then it can propagate. And we also actually deleted polyQ strain. And let's see what I'm going to say. And yeah, it's more synchronous. I'm sorry, I forget what I'm going to say here. It's a pretty, so we got this data like, I don't know, 3 or 4 days ago. So it's not on my head top of my head right now, but
Are they healthier or less healthy?
They are healthier, but the distance between nuclei are a little bit longer. So now I can remember. So in wild type, you can see that all the nuclei are like almost evenly distributed. Like the distance is 3 to 4 micron. But when you delete the polyQ region, then you can now see that some of the nuclei are really clustered, really close in a close proximal. But some of them are really far apart. So we think that in the wild type condition, if you have asynchrony, you can have nuclei pretty evenly distributed. So getting some kind of useful proteins express the transcripts evenly, or get the proteins pretty equally throughout the cell. Does that make sense?
So I was just wondering if you have any evidence that V3 is dynamic, because presumably, after CLIN3 is expressed, it needs to move to a different nucleus.
That is really a question, really good question too. So now we are doing some FRET, the bleaching experiment, and the FCS to see that what's the dynamics in a wild type V3 and a delta polyQ strain, and also like other cytoplasmic GFP. So we clearly saw that the V3 has a really slow, significantly slow diffusion rate compared to the free GFP. And delta polyQ, we're still getting the data, so we don't know yet. But we're expecting to see that seeing a more dynamics in delta polyQ version of V3.
My question's really quick. Is the level of the CLIN3 transcript really the same in the V3 deletion as the wild type? I mean, when it declusters, you see it looks like the levels actually go up, and I can't tell if it's a declustering or.
Yeah, so it is possible that, so I didn't really put it here, but it is sort of natural that, I mean, there are a lot of RNA binding protein that involved in stabilizing the transcripts. And it seems like you see a lot less transcripts in V3 deletion strain. So the density could be the issue, right? But our analysis, the Ripley's K function, actually in here, this is a density independent. So we can just see the spatial pattern. And also delta polyQ has the same really comparable amount of transcripts there, but still shows the asynchronous, the random distribution of transcripts. So density isn't an issue for us.
Sorry, I had a question over here.
Sorry.
Hi. So I guess my question was related to hers. I was wondering if you've looked at the stoichiometry of the V3 and CLIN3 copy number. I'm just wondering if CLIN3 is the only mRNA that V3 binds to, and if it really is that specific, what the basis of that specificity is, if it's sequence or structural or both.
So the V3 actually recognize the UGCAU, and there are like more than 100 transcripts can interact with the V3. And CLIN3 is one of the top transcripts that easily bind with the RN domain in the V3. But there's another possibility of the, any kinds of transcripts or some kind of protein. Like non-cell cycle related transcripts can be bound on there. And we actually found one interesting thing that in V3 deletion, they don't make any branches anymore. They just grow one direction till the end. And delta polyQ too, they just hard to make branches. They're just going up. And we think that it's possible that and we saw that V3 is clustering on the nascent branches. So it's possible that the V3 is also tethering some transcripts related with generating the branches and localize them and concentrate them and help making branches. So there's a lot of possibilities there. And this is one of the.
Have you done any studies to see if the polyQ tract influences the substrate selection for the RRM domain?
No, we haven't. But I actually did a quick bioinformatical research on that, and quickly looked at pull up all the RNA binding proteins in Saccharomyces cerevisiae, and saw that if they have polyQ or if they can be predicted as a prion protein. And very interestingly, 13 or 14 out of 16 or 17 were having the polyQ region, or at least predicted as the prion protein, so it has an unstructured protein structure. So it is possible that they have some kind of connection or utilize one or the vice versa. So, but I haven't seen any kinds of a paper that really study deeply about those things.
Okay, so I think I want to thank all the speakers for lots of thought.